I am Dr. Deepika Malik. Today we are here to discuss the topic electrophoresis. The term electrophoresis describes the migration of a charged particle under the influence of an electric field. Many important biological molecules such as amino acids, peptides, proteins, nucleotides and nucleic acids possess ionizable groups and therefore at any given pH exist in solution as electrically charged species either as cations or anions. Under the influence of an electric field, these charged particles will migrate either to the cathode or to the anode depending on the nature of their net charge. Coming to the types, electrophoresis units are available for running either vertical gel systems or horizontal gel systems. Vertical gel system. A vertical system utilizes a discontinuous buffer system where the top chamber contains the cathode and the bottom chamber contains the anode. A thin gel made of acrylamide is poured between two glass plates and mounted so that the bottom of the gel is submerged in buffer in one chamber and the top is submerged in buffer in another chamber, as you can see in the diagram. When the current is applied, a small amount of buffer migrates through the gel from top chamber to bottom chamber. These systems are ideal for running polyacrylamide gels for SDS Page, which we'll discuss in detail. Whereas in horizontal gel electrophoresis, a gel is cast in a horizontal orientation and submerged in running buffer within the gel box. The gel box is divided into two compartments with agarose gel separating the two. An anode is located at one end while the cathode is located at the other. The ionic running buffer allows for a charge gradient to be created when a current is applied. In addition, the buffer serves to cool the gel, which heats up as a charge is applied. Acrylamide cannot be used for horizontal systems because gels are cast in a tray which is exposed to atmospheric oxygen. Oxygen inhibits the polymerization of acrylamide and thus interferes with the creation of the gel. Therefore, agarose gels are used for horizontal systems such as agarose gel electrophoresis. It is an ideal choice for most DNA and RNA applications. Now let's discuss the vertical gel system, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis or SDS Page in detail. SDS Page is an analytical method used to separate protein mixtures based on their size. The technique is based upon the principle that a charged molecule will migrate in an electric field towards an electrode with opposite sign. Since the mobility of a protein in the gel depends on both charge and size, therefore the protein samples are treated so that they acquire uniform charge and their electrophoretic mobility depends primarily on size. For this, different protein molecules with different shapes and sizes needs to be denatured, done with the aid of SDS so that the proteins lose their secondary, tertiary or quaternary structure. The proteins being covered by SDS are negatively charged and when loaded onto a gel and placed in an electric field, it will migrate towards the anode, that is positively charged electrode, and are separated by a molecular sieving effect based on size. Smaller proteins migrate faster due to less resistance from the gel matrix. After the visualization by a staining protein-specific technique, the size of a protein can be calculated by comparing its migration distance with that of a known molecular weight ladder or marker. The method is called SDS Page as it uses the following two as main components. First is polyacrylamide gel, also called Page. It is used as a support medium. In the experiment, two gel systems with different concentrations are used. First is stacking gel, which forms the upper layer. It is a macroporous gel with low concentration of up to 4%. Buffer used for the formulation of this layer is tris HCl with pH 6.8. Second is the separating gel or electrophoresis gel or resolving gel, which forms the lower layer. It is a microporous gel with high concentration of up to 10%. Buffer used for the formulation of this layer is tris HCl with pH 8.8. The another component for this system is SDS, that is sodium dodecyl sulfate. It is an anionic detergent that is used to denature the proteins. Its use eliminates the influence of structure and charge, and proteins are separated solely based on polypeptide chain length. It is also known as sodium lauryl sulfate. So important points to remember here are that solution composition and pH are different for the preparation of the two gel systems and are also different from the composition and pH of electrophoresis tank buffer. Equipment for SDS Page. Glass plates are used in pair. Spacer or silicone tubing have a thickness of 0.75 mm or 1.5 mm, which determines the loading capacity of the gel. For pouring the gel solution, the plates are usually clamped in a stand with the two spacer on both sides. Binder clips are attached to both the sides of the gel plates to hold the assembly tight. A comb is used to make lanes to load samples. An appropriate comb is used depending on the sample size. For example, we use a 8-lane comb for 7 samples and molecular weight markers. Gel box or tank or chamber. The gel and buffer is placed in an electrophoresis chamber that is connected to a power source and which is designed to conduct current through the buffer solution. Lid is used to cover the chamber. The positive and negative electrodes, which are attached on top and bottom of the chamber and wired through power cables to connectors to allow the connection to the power supply. To apply an electrical field to the gel, an electrophoresis power supply is needed. These power supplies are specifically manufactured for electrophoresis application and features very stable voltage and current outputs to prevent fluctuation in migration speed. The next slide shows different steps of SDS Page. The first step includes sample preparation. Second step includes gel preparation, that includes both separation or resolving gel and stacking gel preparation. Third step is the sample and markers loading. Then we have buffer preparation, power supply, gel recovery, staining, and finally protein extraction from the gel. The diagram here shows a sample that contained two different types of proteins, one with single subunit and the other with two subunits. On treating the sample, the folded structure of the protein unfolds and becomes linear chain of polypeptides. After the sample has been prepared, next step is the gel preparation and then finally the loading of the sample into the gel. After providing the power supply, the sample gets separated based on their molecular weight, which can be detected by staining. Now let's discuss all these steps one by one in more detail. First is the sample preparation. For this, loading or sample buffer is prepared by adding the following components, that includes sodium dodecyl sulfate, also known as SDS, which acts as a denaturant, beta-mercaptoethanol, which acts as a reducing agent, bromophenol blue, that acts as tracking dye, sucrose or glycerol for adding density. Sample buffer is added to protein samples and mixed by flicking the tube. Samples are heated at 100 degrees Celsius for 5 minutes in a water bath. Then centrifugation is done at 15,000 rpm for 1 minute at 4 degrees Celsius. The supernatant obtained contains the protein sample for loading in the stacking gel. Now to understand what exactly these components do, let's review about the protein structure. Proteins are large biomolecules consisting of large chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups along with the side chain. Proteins are formed by linking amino acids with peptide bonds and fold into one or more specific conformations driven by a number of interactions such as hydrogen bonds, hydrophobic interactions, ionic bond and disulfide bond, as you can see in the diagram. The next slide shows the detail about sodium dodecyl sulfate or sodium lauryl sulfate. It is an anionic surfactant and contains a polar head group with a net negative charge at the end of the long hydrophobic carbon chain, as you can see in this diagram. This SDS denatures the native protein by disturbing the hydrogen bonds, hydrophobic interactions and ionic bond. This is very important. While the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, when the protein mixture is heated, SDS binds uniformly to the protein and the intrinsic charge of the protein becomes negligible when compared to the negative charge contributed by SDS. As you can see in the diagram here, this SDS binds uniformly to the protein, making the intrinsic charge of the proteins negligible. Each protein in the mixture is therefore fully denatured by this treatment and opens up into a rod-shaped structure with a series of negatively charged SDS molecules which repel each other along the polypeptide chain, as you can see in this diagram. Now, on average, one SDS molecule binds for every 2 amino acid residues. Therefore, these proteins can now be separated on the basis of their molecular weight. They are strongly attracted towards an anode, that is positively charged electrode in an electric field. Beta-mercaptoethanol is a reducing agent. It is used to cleave the disulfide bonds in the protein structure, as we have discussed earlier also. Bromophenol blue is used as an electrophoretic color marker. It has a slight negative charge and will migrate the same direction as protein, allowing the user to monitor the progress of molecules moving through the gel. Due to relatively small molecule size of bromophenol blue, it is able to migrate faster than proteins. By monitoring the migration front, as you can see in the diagram, the electrophoresis can be stopped before the dye and samples have completely migrated through the gel and leave it. So this bromophenol blue, because of its color, makes easy for us to know when we are supposed to switch off the power supply, because if we don't, the sample will eventually run out of the gel. Sucrose or glycerol gives the sample solution density, thus allowing the samples to settle easily through the electrophoresis buffer to the bottom when injected into the loading well. Now, after the loading gel is prepared, we'll add the sample in it. The next step involves preparation of gel. The formation of gel requires the following components. First is acrylamide or acrylic amide. It is an organic compound with the chemical formula as shown here. It forms linear polymers. Acrylamide gels can be made with a content of between 3% and 30% acrylamide. Then we have bisacrylamide, also known as N,N-methylene bisacrylamide. Bisacrylamide is essentially two acrylamide molecules linked by a methylene group and acts as a cross-linking agent for polyacrylamide gel. The elongating polymer chains are randomly cross-linked by bisacrylamide, resulting in a gel with characteristic porosity. The porosity of the gel is related to the ratio of acrylamide and bisacrylamide. Low concentration of usually 4% is preferred for high molecular weight samples. Now, the polymerization of acrylamide is an example of free radical catalysis and is initiated by the addition of ammonium persulfate or APS. The polymerization reaction is initiated by a free radical generating system. APS is used as a free radical initiator. These free radicals convert acrylamide monomers to free radicals which react with inactivated monomer to begin polymerization chain reaction. Timid, that is N,N,N,N tetramethyl ethylene diamine. It catalyzes the formation of free radicals from APS. Timid accelerates the polymerization of acrylamide and bisacrylamide. Photopolymerization is an alternative method that can be used to polymerize acrylamide gels. The ammonium persulfate and timid are replaced by riboflavin, and when the gel is poured, it is placed in front of a bright light for 23 hours. Photodecomposition of riboflavin generates a free radical that initiates polymerization. Now, the first type of gel is separating or resolving gel. Acrylamide solution of higher concentration of about 10% is poured firstly for a separating gel, normally about 5 cm long, using an autopipette. It is placed vertically at room temperature. It forms the lower layer. Buffer used for the formulation of this layer is tris HCl of pH 8.8. It is overlaid with water to prevent contact with air, which inhibits polymerization. Acrylamide is allowed to polymerize for 20 to 30 minutes to form a gel. The overlaid water is removed after it polymerizes. The sample to be separated are not in fact loaded directly into the main separating gel. Proteins migrate at different rate depending on the concentration of the separating gel. Use an appropriate gel concentration for your target protein. Using a higher acrylamide concentration produces a gel with smaller mesh size, that is microporous, suitable for the separation of small proteins. In general, an acrylamide concentration between 6 and 15% is used. Gels with an acrylamide concentration gradient, that is gradient gels, are also used. As you can see in the diagram, we have a gel between the two glass plates and the separating gel forms the bottom part of the gel. Now for the stacking gel, acrylamide solution of lower concentration of about 4% is poured over the separating gel for making a stacking gel approximately 0.8 cm long. A comb is then inserted immediately to avoid air bubbles and the gel is allowed to polymerize. After the gel is set, binder clips, spacer and comb is removed from the gel assembly. The purpose of this stacking gel is to concentrate the protein samples into a sharp band before it enters the main separating gel. The purpose is to ensure that all proteins arrive at the separating gel at the same time because gel wells are around 1 cm deep. So in the absence of stacking gel, all the proteins will enter the separating gel at different times, resulting in very smeared bands. The stacking gel has a very large pore size which allows the protein to move freely and concentrate or stack under the effect of the electric field. Now let's see what is running buffer. Once the gel is completely polymerized, the gel cassette is placed vertically between two electrodes into a chamber, as you can see in the diagram. Positive electrode is located at the bottom of the gel, whereas negative electrode is located at the top of the gel. The tris glycine chloride electrophoresis buffer of pH 8.3 is poured in the tank to allow for conduction of current through the gel. SDS is also present in the running buffer to make sure once the proteins are denatured, they stay that way throughout the run. Chloride ions are negatively charged, while glycine can exist in 3 different charged states depending on the pH. The isoelectric point of glycine is 5.97. At this pH, glycine carries neutral charge. At a pH below its isoelectric point, glycine carries a net positive charge, and at a pH above its isoelectric point, it carries a slight negative charge, as you can see here in the diagram. The table here shows the running buffer components that includes tris, glycine, SDS, deionized water and a pH of 8.3. Now by using a micropipette, the prepared protein sample is carefully loaded into the wells of the gel, as you can see here in the diagram. To ensure accurate quantification and reproducibility, it is essential to load the same amount of protein for each sample. Further, to assess the molecular masses or sizes of proteins in a gel, a prepared mixture containing several proteins of known molecular masses is run alongside the test sample in one or more lanes of the gel. Such sets of known proteins are called protein molecular weight or mass markers or protein ladders. A standard curve can be constructed from the distances migrated by each marker protein. The distance migrated by the unknown protein is then plotted and the molecular weight is extrapolated from the standard curve. After the loading, turn on the power supply and run the gel until the dye bromophenol blue in the sample buffer reaches the bottom of the gel. Now the next slide will give you a clear understanding of how proteins are being separated within the gel. The first diagram here shows just one well. This well contains proteins, chloride ions and glycerine. The chloride ions and glycine are basically part of the running buffer. So these proteins and chloride ions are negatively charged here. The running buffer, which has a pH of about 8.3, which is above the isoelectric point of the glycine, that is 5.97, makes the glycine to have a negative charge. Now in the second diagram, once electric current is applied, the proteins, glycine ions and chloride ions are forced to enter the stacking gel where the pH is 6.8. Now in continuation of this, the third diagram shows the stacking and separating gel together. The pH in stacking gel is almost similar to the isoelectric point of glycine. Therefore, at this pH, glycine switches predominantly to its neutral state from the negative charge. This loss of charge causes glycine to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride migrate in front of the protein as leading ions and glycine move behind the proteins as trailing ions. Consequently, the proteins are trapped and concentrated in a narrow zone between chloride and glycine molecules. Now this type of movement is carried on until it hits the separating gel where the pH changes to 8.8. At this pH, Glycine molecules are mostly negatively charged and can migrate much faster than proteins. Proteins are now separated, so high molecular weight proteins move more slowly through the gel than low molecular weight proteins, and the separation is achieved. After this, remove the gel assembly from the electrophoresis apparatus. Remove the gel from the glass plates using a clean spatula and discard the stacking gel. After this, place the separating gel in a plastic tray containing gel fix solution, which is a methanolic solution acidified with acetic acid. The proteins are fixed by acetic acid and then stained using Kumasi Brilliant Blue. It is the most popular protein staining dye. It is an anionic dye which binds non-specifically to proteins. The Kumasi dyes R250 and G250 bind to proteins through ionic interactions between disulfonic acid groups and positive protein amine groups, as well as through van der Waals attractions. After staining, the proteins are detected as blue bands on a clear background, as you can see here in the diagram. After visualizing the protein, it is often necessary to elute and recover proteins separated by SDS Page. The procedure involves localizing the protein of interest on the gel following SDS Page, then eluting the protein from the gel, removing SDS from the eluted sample by acetone precipitation, and finally renaturing the protein, enzymes for example, for subsequent analysis. Protein yields ranging from nanogram levels to 100 micrograms have been obtained. Proteins are extracted or eluted from the gel by several methods. These includes dissolution of the gel matrix, that dissolves the gel matrix by incubating the gel matrix in 30% hydrogen peroxide at 50 degrees Celsius. Passive diffusion, water or buffer is added to a piece of gel containing the protein of interest. Trapped protein molecules diffuse out of the gel matrix into the surrounding medium. Electrophoretic elution. In this technique, protein containing gel pieces are placed in an electroelution chamber where the proteins are eluted from the gel matrix into a buffer solution using an electrical field. Now coming to the applications of SDS Page. It is a widely used technique in biochemistry and molecular biology and is employed in various research areas and applications. First is protein purification and characterization. It is often used in protein purification workflows to assess the purity of protein samples. It helps to determine the molecular weight of the target protein and confirm its presence in the purified fractions. Additionally, SDS Page can be used to assess protein stability, monitor protein degradation, and study protein-protein interactions. Protein expression analysis. SDS Page is used to analyze protein expression levels in different samples. By comparing the intensity of protein bands between samples, researchers can determine relative protein abundance and identify proteins that are upregulated or downregulated under specific experimental conditions. This information is valuable for studying protein functions, cellular responses, and disease mechanisms. Western blotting. SDS Page is an essential component of the Western blotting technique. After proteins are separated by SDS Page, they are transferred onto a membrane and probed with specific antibodies to detect the presence of a particular protein. This technique allows for the detection and quantification of target proteins in complex mixtures and is commonly used in protein research, diagnostics, and biomarker identification. Then protein size determination. SDS Page enables the estimation of the molecular weight of proteins based on their migration through the gel. By comparing the migration distance of unknown proteins to that of known molecular weight markers, researchers can estimate the size of the target proteins. This information is critical for studying protein structure, function, and interactions. Protein complex analysis. This technique is used to investigate protein-protein interactions and analyze the composition of protein complexes. By performing SDS Page under non-denaturation conditions, native protein complexes can be preserved and their subunit composition and stoichiometry can be determined. This helps in understanding the organization and dynamics of protein complexes and their role in cellular processes. Peptide mapping. SDS Page is commonly used in peptide mapping, a technique that involves the digestion of proteins into peptides and the subsequent analysis of those peptides. Peptide mapping is used to identify protein sequences, study post-translational modifications, assess protein integrity, and evaluate the similarity between protein samples. Quality control of protein-based products in biotechnology and pharmaceutical industries. It is used to assess the purity, integrity, and stability of recombinant proteins, antibodies, vaccines, and other biologics. SDS Page provides valuable information about the presence of impurities, aggregation, and degradation product, ensuring the consistency and safety of these products. So with this, we wind up the principle and application of SDS Page. In the next session, we'll discuss agarose gel electrophoresis.